Good afternoon. This is for A&P Lab for North Dakota State for the paramedic program. And today we're going to be dissecting a sheep eye. It's also very similar to the structures of a cow eye as well. Something to note right on the exterior portion of this eye is the cornea. Here in death and with a lot of preservatives, it's clouded, but in life it's actually quite clear. And something to note in cows and sheep as well is that their pupil is actually horizontal, where us humans have a round pupil. There's also the sclera, which is the white of the eye. That's going to be a little easier to see once we open that up. And then we also have our optic nerve on the exterior side here, on this posterior portion. In this particular specimen, it's quite small, quite short, of course, where it's been severed. But that optic nerve is responsible for transmitting and receiving information between the eye and the brain. That nerve is also the cranial nerve 2. So we've made a cut along the coronal plane, which separates both the anterior and the posterior portion of the eye, and which we'll notice here when we open up those two hemispheres is right off the bat, we'll notice that sclera, that white portion of the eye. And the sclera is what's responsible for that durability. It's that connective tissue that protects a lot of these inner structures. It helps maintain a lot of rigidity, and it's really keeps things protected in there. It's a little bit torn apart here, but you can see some of the retina that's remaining. The retina has a consistency almost like wet tissue paper, so once it's all clumped up, it's really difficult to get spread back out. But that retina, that's that light sensitive portion of the eye. And if you recall rods and cones, that's the receptor cells, the bundle of nerves that makes up this retina. And those rods and cones, those receptor cells, are responsible for how we perceive light intensity. The rods, of course, are responsible for more dim lighting, evening lighting, while the cones, they're more responsible for color and the daylight. And there is a blind spot here in this retina. If this were spread out, this is, again, a very light sheet that's separating this or covering this particular portion of the eye. And there is a blind spot that will connect there and lead ultimately, connect at the optic disc and lead out through the optic nerve. And in that blind spot, our brain compensates for this, and it's not something we even realize, but there's only axons in that. There's no receptors, and they meet up at that particular point there at the optic disc that then goes out to that optic nerve on that posterior portion. But normally, again, this retina has like a tissue paper consistency, would be spread out along this particular plexus here. And this here is known as the choroid. That's a dark sheet. You can see that layer that comes up a little bit there. That dark sheet of tissue is between the retina and the sclera, between that white portion there. And that provides a lot of nutrients for the inside of the eye and around the eye. And something that's unique to mammals, in particular deer and sheep and cats as well, is this iridescent coloring, this beautiful blue and green that humans do not have. And this particular part of the choroid tissue helps in night vision by reflecting any kind of available light back into the retina. So this, again, is not present in humans, but in sheep as well as other mammals. So that's a really unique structure here. When we first opened this up, we have a lot of aqueous humor. Most of this is a clear fluid. A lot of the preservatives, formaldehyde, have gotten in here and seeped in here, and of course is responsible for a lot of that dark coloring. But that aqueous humor is composed mostly of water, and it helps maintain the pressure and the shape around that eyeball. And so once we look on the anterior portion here, you'll notice the lens is surrounded by a pretty gelatinous tissue, and that is called the vitreous humor. It's very gelatinous, helps maintain the shape of the eyeball as well, and that's found here in the vitreous body. That's a cavity between the retina and the back of the lens, and that holds the lens, which is a very tough, hard structure. It's the one structure where it's pure protein fibers. And in my research, I found that it's the only part of the body that actually is not susceptible to cancer. So that's a pretty interesting little tidbit there as well. If we go a little bit further, you'll notice the pupil, which is simply the opening, which allows the light into the eye, and the diaphragm, which the iris, which is the diaphragm that regulates the size of the pupil. So when we look a little bit more closely here, it's almost like inside of a mushroom cap. There's different ciliary bodies and muscles that hold all of this together. These suspensory ligaments, that's what controls the size of this lens, holds that lens in place. So that's pretty interesting as well. We also have the hyaloid fossa, and that's that indentation here in this vitreous body that's going to support this lens. The other thing to note too is the zonula solaris, which is the ligaments that suspend and hold the lens in place. Helps us focus on vision by stretching, dilating, and constricting. One more portion of the sheep eye to note, again, is that tapetum lucidum. That's that iridescent portion. But other than that, that pretty much covers the sheep eye. Pretty interesting structure. I did remove a lot of the tissue on the outer ends of it. There were some muscles there, but mostly fat tissue that surrounds that particular portion of the eye. Muscle, fat, connective tissue, again, provides a lot of nutrients and support and structure around that eye as well. And that completes the sheep dissection for A&P, North Dakota State. Thank you.